have you on Planetterrian Daily, episode 75 for June 1st, 2026. Some interesting findings to cover today. AI analysis of CT scans links healthier thymuses to longer lifespans and lower disease risk in adults. Researchers at Mass General Brigham used AI to examine CT scans from tens of thousands of adults. People with healthier thymuses showed longer survival and substantially lower risks of heart disease, cancer, and death. This small immune organ, once viewed as largely inactive after childhood, now appears tied to broader aging outcomes. The finding adds a measurable imaging marker to ongoing efforts tracking cellular aging biomarkers. Next steps include testing whether thymus imaging can refine risk models in longitudinal cohorts. The thymus imaging study from Mass General Brigham adds a concrete, non-invasive marker to aging biology research. Healthier thymuses correlated with lower rates of heart disease, cancer, and overall mortality across tens of thousands of adults, moving beyond the long-held view that the organ becomes irrelevant after childhood. This finding sits within the tracked program on aging biomarkers and raises whether routine CT analysis could eventually stratify risk or guide interventions that preserve thymic function. What remains unclear is how modifiable the thymus remains in midlife and whether imaging changes track with other hallmarks of cellular aging. Which existing longevity cohorts could add thymus metrics to test these links? Most people assume the thymus shrinks into irrelevance after puberty and plays no further role in adult health. In reality, the organ continues to export T cells and support immune surveillance well into later life, even if at reduced volume. Right now, as you sit reading this, your thymus is still shaping the diversity of T cells circulating through your blood and tissues. AI analysis of CT scans from tens of thousands of adults showed that individuals whose thymuses retained more tissue volume lived longer and experienced markedly lower rates of heart disease and cancer. The memorable detail is that this single imaging feature tracked with broad survival differences across a large population, independent of obvious clinical markers. One practical takeaway is to ask whether future routine scans could flag thymic health the same way they already flag bone density or vascular plaque, prompting earlier attention to immune resilience. From internal immune organ imaging to another electrophysiological signature that distinguishes conscious states, we turn next to a newly identified brain rhythm. Neuropsychology researchers identified a distinct midbrain oscillation present only during waking hours and REM sleep. The rhythm is absent in other sleep stages, positioning it as a potential biophysiological signature for specific consciousness levels. The work maps neural dynamics that distinguish alert and dreaming states from deep non-REM sleep. It supplies a new electrophysiological target for studies of brain aging and neurodegeneration. Follow-up experiments will test whether the signature shifts with age or disease. While that rhythm helps map brain states, another technology now lets researchers both record and control large neuronal populations at once. Nature Methods describes NeuroPixel's optoprobes that combine high-density electrodes with optical waveguides for blue and red light delivery. The devices record spikes from large neuronal populations while activating or silencing defined groups via optogenetics. Mouse brain tests showed clean separation of recording and stimulation sites. The single shank design simplifies experiments that previously required separate tools. Expanded use in chronic preparations is the next technical step. From probing living brains, we move to mapping the proteins that build them, where scale has just increased dramatically. A new iteration of protein folding software has generated predicted structures for 1 billion proteins. The expanded database supplies atomic-level models for proteins previously lacking experimental data. Researchers can query the set to identify folds linked to disease or to design new enzymes. The scale moves structural biology from individual targets toward proteome-wide analysis. Integration with experimental validation pipelines is the immediate next phase. With structures in hand, the next question is how cells use them to self-assemble tissues without external direction. A perspective in Nature Biotechnology frames cellular self-organization as a core mechanism underlying multicellular life. Stem cell-based models are proposed as tools to map how cells spontaneously form tissues without external scaffolds. The framework connects developmental biology to regenerative engineering goals. It highlights safety questions around partial reprogramming that remain open in the field. Ongoing model refinement will test whether these principles scale to organ-level constructs. Refining which cells carry precise edits remains a practical bottleneck. A new selection method addresses that directly. Nature Biotechnology reports a transient AND gate reporter system that selects human hematopoietic stem cells carrying intended gene edits. The approach raises the fraction of correctly modified cells after homologous recombination. It reduces the burden of off-target or unedited cells in therapeutic preparations. The method targets a key bottleneck in ex vivo gene editing for blood disorders. Clinical translation will depend on scaling the selection step without compromising cell viability. New imaging and probe technologies continue to sharpen how we observe both the body's internal clocks and the universe beyond. Before we go, keep an eye on scaling efforts for the AND gate selection system in hematopoietic stem cell therapies. That covers today's science and health news. Share this with someone who's curious about the latest research. I'm Patrick in Vancouver. See you next time. And before you go, this show is part of the NERA Network, a family of daily podcasts covering tech, science, markets, and more. If you enjoyed today's episode, give Environmental Intelligence a listen, the Environment and Climate Policy Brief for Canada. You can explore the whole lineup at neranetwork.com. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.